Hello, this is Mary, and this short video is to talk about CRISPR. Perhaps you've heard of CRISPR as the biotechnological tool that allows us to edit genomes, to inactivate single genes, or to replace individual genes. This is miraculous and a very specific phenomenon. Imagine you have a book that is 6 million letters long and you have the capacity to go in and change one single letter, find and change one single letter. And that's what the CRISPR tool allows us to do. But what I want to talk about today is I want to talk about the biological phenomenon that is CRISPR. Let's talk about the acronym. The acronym CRISPR stands for clustered, regularly interspersed, short palindromic repeats. These CRISPR sequences in the genome of bacteria were identified as being responsible for what is called acquired immunity. You're likely familiar with acquired immunity as what happens subsequent to infection or vaccination against a human pathogen, for example. Your immune system can store the memory of that infection in B and T cells and will have an army of responders at the ready for a time should a subsequent reinfection occur. Well, bacteria have a similar phenomenon. Since they're not multicellular, they store all the equipment in their genome. So let's sketch a bacterial cell. Let me go ahead and depict the genome of that bacteria, which is circular, as are all bacterial genomes. Within that circular genome are found clustered, regularly interspersed repeats, and I'm going to depict them with this purple marker here. They are repetitive. They are also palindromic. What that means is the sequence of the top strand is identical to the sequence of the bottom strand backward. Interspersed between those repeats, scientists found a number of different sequences, all unique, that bore sequence similarity to bacterial viruses. Bacterial viruses are called bacteriophage. So the bacterium is storing in its genome a representative sequence from dozens or hundreds of different bacteria, which it has different viruses, which it has previously been infected with. Let us imagine that this bacteria subsequently encounters a virus that contains this green sequence. So let's say this is the green bacterial virus, and it's going to put its DNA into the bacterial cell, ready to hijack all the protein synthesis equipment in the bacterial cell to make lots and lots of copies of itself and destroy the bacteria while it's at it. However, the bacteria has a defense if it has seen it before and stored its sequence in the CRISPR array. What it will do is it will copy the sequence of indeed all of the viral sequences in the array. It will copy this green one and it will attach to it a special protein that is a nuclease. A nuclease is an enzyme that destroys nucleic acid. So what is going to happen is that this Cas9 guide RNA complex, we call this RNA here the guide RNA or gRNA, it will find the complementary DNA or RNA from the bacteriophage or virus, bring the molecular scissors to it, and this is my depiction of a scissors, and it will destroy or break up all of that invading viral nucleic acid, effectively disarming the pathogen. So what we're going to learn about later and in another video is how, because of the specificity of the detection of the viral RNA by the guide RNA, we are going to take advantage of that. It's very specific because it uses nucleic acid complementarity, which is very specific. And we're going to take advantage of that to allow us to target a Cas9-like enzyme to a specific spot in a genome representing a gene in an organism, perhaps a gene we want to destroy or a gene we want to replace. But more about that later. But for now, this is the biological phenomenon that is CRISPR. Thank you for your attention.